So today we will talk about the most important topic of our chapter chromosome and DNA, which is regulation of gene expression. Basically, gene expression means what? The conversion of DNA into mRNA and then mRNA basically into protein, right? As we already know that every cell needs protein, right? Every cell requires protein to do to do its functions. Basically इसका पूरा structure बनाने के लिए हम सेल को उसका स्ट्रक्चर बनाने के लिए और बेसिकली उसके अंदर जो सेल डिफ्रेंसिएशंस होनी है उन सब चीज़ों के लिए प्रोटीन की ज़रूरत है. तो उसके लिए हमारे सेल के अंदर बेसिकली हमारे जीन के अंदर एक्सप्रेशन होती है. एक बेसिकली रेगुलेशन ऑफ जीन्स का प्रोसेस होता है हमारे जीन्स के अंदर. बेसिकली अगर हम फॉर एग्ज़ाम्पल हमारे एक सेल को XYZ प्रोटीन चाहिए तो जाहिर सी बात है हमारा सेल हर वक्त उस जीन और उस प्रोटीन को तो नहीं बनाता रहेगा. वी आर सेल वर्क्स ऑन इट्स रिक्वायरमेंट कि उसे किस वक्त किस प्रोटीन की रिक्वायरमेंट है. तो फॉर दैट प्रोसेस बेसिकली द ऑन एंड ऑफ जीन मैकेनिज्म इज प्रेजेंट. द ऑन and off gene on and on mechanism of expression. Basically this on and off gene mechanism is present so that our cell can work on its requirements. If for example this XYZ protein is required at that moment, so our genes will be focused on making that gene on that protein. If it is not over there, so it means our cell will be continuously working on this XYZ protein. So this is not how our cell can work, right? For example, basically this regulation of the gene expression is done by metabolic, environmental and some physiological conditions. For example, if we talk about E. coli is an basically a bacteria and it is cultured. If we are doing its culturing, consider we have two mediums here, right? The first one is if we are having the lactose medium, first one, and second is like we are not using any kind of medium, so it is no medium, right? So in lactose medium, for example, in order to make the lactose is what? Lactose is disaccharide. So for that an enzyme is used which is known as beta galactosidase. It is an enzyme which is used to make this lactose. So lactose is what? Lactose is basically a disaccharide, right, which is made up of glucose plus lactose. So in order to make this lactose, basically there is a gene turn on mechanism. So the genes of basically this beta galactosidase are turned on. Genes are turned on. But on the other hand, if we see that we don't have any kind of medium, so it means that we don't have any metabolic reaction. There are no metabolic reaction, right? It means that no enzyme synthesis. It means that it means that genes are turned off. Genes are turned off, right? So if we talk about this was E. coli is for E. coli bacteria. We are talking about the prokaryotes, right? But if we talk about the users, if we talk about the multicellular organisms, right? If we talk about multicellular organisms, right? So basically a process of cell differentiation takes place in them. Cell differentiation. How? For example, consider that we have meristematic. Consider we have meristematic. Sorry. We have meristematic tissues. Basically, they have the capability to divide. They are basically dividing tissues. Right. Now these tissues are basically converted into permanent tissues. This process is known as differentiation. This process is known as differentiation. It means that if now they have become permanent tissues, it means they have basically loosened the property of division. They have loosened the capability of division, right? Now basically now these permanent tissues are again converted into meristematic tissues. Meristematic tissues, and this process is basically known as dedifferentiation. It means now they have regained the property of division. Regain the capability of division after losing it. After losing, right? Now these meristematic tissues are again converted into permanent tissues, and this process is known as redifferentiation. Right, redifferentiation is what? Basically it is losing after regaining. Losing what? Losing the property of division after the regaining. So this is what? This is basically cell differentiation. It happens in the multicellular organisms, right? So now let's discuss like what if this on and off mechanism is absent in our body, in our cells. Like you can see that there can be a lot of issues in our body. Basically, it can lead to cancer as well. For example, you can see that there is abnormal growth of some cells, right? So basically this abnormal growth is what? It is basically tumor. So if there won't be any kind of mechanism of gene regulation, the gene turn and off, so this tumor will lead to cancer. Even some lysosomal disorders can also take place. Lysosomal disorders can also take place. For example, lysosome storage disease. Lysosome storage disease, right? For example, okay, now let's talk about some importance of this gene regulation mechanism. What is the importance of this turn and on and turn off mechanism, right? So what is basically the importance? Okay, if consider that basically this gene regulation mechanism conserves our energy, and it also conserves space. It conserves energy, plus it saves the space. Now, how it basically conserves energy? For example, there are certain proteins that we don't need anytime, every time, right? अगर हमारे पास जीन रेगुलेशन अगर मैकेनिज्म अगर नहीं होता तो मतलब जो जीन्स हैं वो तो सारा दिन बनते ही रहते, वो हर टाइम अपनी प्रोडक्शन करते ही रहते, ड्यू टू दिस जीन टर्न एंड ऑफ मैकेनिज्म हमारे पास ये जो प्रोटीन्स है ये सारा दिन बनते नहीं रहते, एक ही पर्टिकुलर टाइम पे बनते हैं। तो इससे क्या होता है, हमारे पास एनर्जी जो कंजर्व होती है, अगर वो हर टाइम बनेंगे तो हर टाइम उनके लिए एनर्जी चाहिए होगी। जाहिर सी बात है जीन्स ऐसे तो काम नहीं करेंगे, उनके लिए एक एनर्जी की पर्टिकुलर ज़रूरत होगी। तो अगर वो हर वक्त काम नहीं करे, इसका मतलब है कि वो एक अपने अंदर एक एनर्जी कंजर्व कर रहे हैं, एनर्जी बचा रहे हैं। बट इफ यू टॉक अबाउट कि स्पेस कैसे कंजर्व हो रही है, कि वी हैव डिस्कस रेप्लिकेशन, आपने पढ़ा होगा। तो जब रेप्लिकेशन के अंदर जब अनवाइंडिंग होती है, जब हेलिकेज एंजाइम और गायरेज एंजाइम मिलकर सुपर कॉलिंग हटा रहे होते हैं, तो एक पर्टिकुलर ओरिजिन साइट पे होती है। अगर हम प्रोकैरियोट की बात करें या यूकैरियोट की बात करें, दे आर पर्टिकुलर ओरिजिनल साइट। सो इन दैट ओरिजिनल साइट द रेप्लिकेशन बिगिन्स। सो इट मीन्स इट कंजर्व्स स्पेस। अगर पूरा डीएनए खुल जाएगा तो जब हम अनवाइंडिंग करते हैं, सो इट मीन्स इट रिक्वायर्स स्पेस। तो जब हम एक पर्टिकुलर साइट पे करते रहेंगे, पर्टिकुलर ओरिजिन वहाँ पर साइज होंगी, तो बेसिकली वो कंजर्व करेंगी स्पेस को बचाएंगे। क्योंकि पर्टिकुलर जगह पे हो रहा है, हर डीएनए का सेगमेंट के अंदर अनवाइंडिंग नहीं हो रही, तो वो हमारे स्पेस को कंजर्व कर रहा है, राइट? दिस इज हाउ इट बेसिकली सेव्स द स्पेस. Now, if we talk about, for example, if we talk about liver, right? If we talk about liver, so we know that liver, what is the function of liver? Detoxification. What it does, basically it converts alcohol into non-toxic substance by the help of alcohol dehydrogenase enzyme, right? Right, but this production of even this enzyme, the production of this enzyme is also is in the hand of this gene regulation, this in a turn on and off mechanism, right? किसी और के पास ये फंक्शन नहीं है कि वो डिटॉक्सिफिकेशन करे, क्योंकि सिर्फ लिवर के अंदर ही ये जीन्स टर्न ऑन है. इफ यू टॉक अबाउट फॉर एग्जांपल आरबीसीज कैरी व्हाट? These RBCs carry oxygen, right? But this liver can't carry oxygen. Why? Because the genes of to carry the oxygen, genes are turned on here, and genes are turned off here. The genes to carry the oxygen are turned on here, but they are turned off in the liver. So basically, this is how the gene regulation, the turn on and off mechanism basically works, right? So I hope this is clear to you. If you have any kind of queries, you can ask me in the comment section. I'm really glad to answer you guys.